Okay, aloha everyone. Thank you for joining us for our weekly Hanauma Bay education program, Hanauma Talks, for Thursday, November 5th. I'll be introducing Maya Walton, who will be introducing our three graduates who will be talking about their research. So, Maya?
Aloha everyone, thank you so much for joining us. I work at the University of Hawaii Sea Grant College program, and in the months of November and December at Hanauma Bay, but this year virtually online via Zoom, we feature the research of Hawaii Sea Grant graduate fellows and PICAST graduate scholars. And we're really excited to have three of our graduate researchers join us tonight to share the results of their work, or the work that's in progress this year. Yeah, so the aim of the program that these Sea Grant graduate fellows and PICAST graduate scholars are in is not only are they engaging in scientific research and helping to advance knowledge in their fields, they're also gaining training in science communication and how to be better community partners. So that's a little bit about our fellows and scholars. So the three people that we're going to hear from tonight, the first person we'll hear from is Becca Lensing. Her talk is titled, "Do Hawaiian Microbes Eat Their Leafy Greens?" The second talk will be from Marcus Pang. His talk is, "What's a Beach Day Worth? Economic Valuation of Waikiki Beach." And last but certainly not least, we will hear from Honor Booth, who will present her talk titled, "Ai Ya Eka Ia, Ai Ya Eke Kanaka, Transfer Pollutants by Fish." And please feel free to enter questions into the chat box as we go along. Gavin and I will be watching the chat, so please put your questions in there. Like Gavin said, if you can mute your microphone, and I can see pretty much everyone has their microphone muted, that helps us a lot. So we'll take a question while we switch in between speakers, but we'll have plenty of time at the end to have lots of questions from the audience. Okay. Becca, the floor is yours when you are ready.
Mahalo. All right, I'm gonna set my own timer here. Thank you. Just a little bit about myself. So I'm a fifth-year PhD student in the Marine Biology graduate program at the University of Hawaii at Manoa in Rosie Allegato's lab. And two disclaimers before we get started today. One is that this talk will sort of take on a research proposal perspective. We've had some unfortunate setbacks with coronavirus, which is just the reality of science, but science always moves forward. And second disclaimer is that I'm a big Star Wars fan, although maybe not as big of a fan as my advisor or even my office mate. So with that, all of you that live here in the Hawaiian Islands, I hope you know that mangroves are invasive to the Hawaiian Islands. They were introduced back in 1902 to prevent sediment runoff onto the reef. And this is a problem because mangroves drastically change their coastal environment. They're what we call keystone species, and typically this is for the benefit of their environments. So in their native systems, mangroves help to protect shorelines. They stabilize the sediments from running off and eroding. They provide a lot of energy and food to the food webs, and they can serve as nursery grounds for native species. In invasive mangrove systems, however, such as the one pictured here on Dagobah, mangroves can trap sediment, trash, and pathogens. If you've ever walked through a invasive mangrove forest here in Hawaii, for example, it's dark, it's it smells, it's very much swamp-like, and the trees, they just tower above you because they can grow up to 80 feet tall. And you take a step and you just sink up to your waist in mud, much like Luke Skywalker's X-Wing, just down into the bog. They can also block water circulation, so wind and tidal forces that would normally stir up the water They can deplete the oxygen and suffocate the fish in the water. They alter the food webs by really changing the base of the food system, and they can serve as nursery grounds for invasive species, which is not good for our native species. And just for comparison, here is a picture of one of the invasive mangrove forests here on the islands of Oahu. So here is a somewhat simplified schematic of the food web in a native mangrove forest. There are two ways that mangrove detritus can be incorporated into the food web. That is either by grazing by native grazers, which have co-evolved with mangroves. So they have these specialized gut microbes that can break down the mangrove detritus. The second way is that this sloppy feeding of the native grazers as they're eating and breaking up the mangrove into bits and pieces. External microbes in the environment like bacteria, fungi, or protists can then further break it down and then get eaten by other organisms in the food web. But what about the Hawaiian food web? You know, we have all these wonderful native species that have never seen mangroves before. You know, they look at it very much as like a child looks at his vegetables. It's just, you know, there's no way I'm gonna eat that. It looks weird, it smells weird, it tastes weird, I'm not gonna eat it. And it's just, it's not good for any of the native Hawaiian invertebrates or fish in the systems. There were some introductions, so we have purposely introduced some species like the Samoan mangrove crab to purposely eat the mangrove, and then we can eat them. But unfortunately, it sort of resulted in the situation of these invasive species don't actually eat the mangrove. You know, why would they want to eat mangrove salad when they can eat polychlorinated poke? Like, Hawaiian food is just so much more delicious. So that really begs the question of, are the microbes still breaking down the mangrove detritus? You know, are they still eating their leafy greens? And so we're going to use a two-pronged approach to answer this question. We'll do DNA sequencing. So we'll take a sample, we'll extract all of the DNA in that sample, we'll break it up into bits and pieces, sequence it on a sequencer, get our A, C, T's, and G's, and do some bioinformatic analysis. And this particular analysis will help us to help the microbes sort of brush off their resumes. And that's a very important thing to do in the time of coronavirus. So using this method, we can sort of ask the microbes, what are your specific traits or abilities? What special skills do you possess? You know, are you a sulfur oxidizer? You don't need to breathe oxygen if you can breathe sulfur. Are you a photosynthesizer? Or are you a decomposer? Maybe you're really great at making the best situation from somebody else's very bad day. Or are you part of a teamwork pathway, for example, for nitrification processes? But of course, just because they possess the potential to do this doesn't mean they actually are. So then we'll also use stable isotope analysis to ask if they really are eating their leafy greens. And this is just a really fancy technique that pins on the basis of you are what you eat. So in the environment, there are different kinds of nutrients, and these nutrients exist both in light and heavy forms. So say, for example, Inky and Pinky here, if you're familiar with Pac-Man, they decide to go out for Pauhana one night. Inky, blue guy, is a very picky consumer. His body is his temple. He's really watching his pandemic pounds. So he preferentially consumes the light nutrients. And then Blinky is a much less picky eater. He will consume whatever you put in his hand. So he consumes the regular nutrient. They don't quite keep their wits about them. They get eaten by two Pac-Men. And then those Pac-Men incorporate what used to be Inky and Blinky into their biomass. And then using stable isotope analysis, scientists, we can actually go back to these Pac-Men and say, this Pac-Man ate Inky because it is made out of the light nutrient. And then this Pac-Man ate Blinky because it is made out of the heavier nutrient. This works in the real world. So say, for example, we take the nutrient carbon. Mangrove is a very picky eater. So it will preferentially consume the lighter forms of carbon as opposed to other primary producers in the system, which generally aren't as picky. So they will incorporate that heavier carbon at a much higher rate. And then we'll also be asking this across a gradient of forest and restoration ages using a space for time sampling process. So if you imagine a forest that you're standing in, and you can walk from the newest, youngest part of the forest, and you can walk through the forest, and as you walk, you're walking to older parts of the forest. And then you reach the end of your path and you're at the oldest part of the forest. So similarly, you know, we will be doing a similar transect, which will become apparent when we look at our sampling sites, but we are able to sample young forests, sample moderately aged forests, and sample very old forests to sort of substitute this space for time. And similarly, along a restoration gradient as well. So that leads me to our sampling sites. First off, Polyaromatic here on the southeast shore, you can actually literally see the mangrove that has infiltrated the shore. It's all of that greenery here in this box. And then you can also see it in a nice aerial view here. So let me walk you through this map. So in green is the mangrove extent as it was back in 2016. Outlined in blue are the fish ponds that existed prior to the introduction of mangrove, including one of the oldest and largest fish ponds in the Hawaiian Islands, which is obviously no longer functional, but that's a palau. And the coastal springs in these are the coastal freshwater springs in the orange circles. Now let me walk you through this map. This map designates the different time periods in which growth was seen on this island. So the older forest here is Mauka, the newer forest here is Makai. And each of these, the lines, the colors represent a different age of forest. So we can literally walk from the old forest to new forest or vice versa. And that's literally what we'll do. We'll go, this is one of the unfortunate things where due to the inter island quarantine and regulations, we're still waiting to do our field work here, but very much looking forward to that. All right, and then our other site is a little bit more local here in Kaneohe Bay in Heia, Oahu. Here's a wonderful picture. Let me walk you through this map. So the line here in red signifies where there is currently existing mangrove. In purple is where mangrove has been removed, and those are designated with the different years that the mangrove was removed. And this is the current removal that they're currently working on for the 2020 removal. And as you can see, this follows along a gradient where essentially they started on the south side of the pond and they removed along the wall in a very sequential and organized way. So in this way, we can sample different aged restoration plots. Like so. So these are some of our proposed spots. And then here is a drone video courtesy of Kali'i Kubite, one of our Pai guys, for some of the removal they're currently doing along this blue strip here, which is pretty fantastic. Okay, so why are we doing this? Why is this even worth discussing? So it's important to keep in mind that forests change as they age. And so it's really important for us to measure different aspects of forest progression here in the Hawaiian Islands, especially since the complete removal of mangrove is unlikely. It grows too fast and it has infiltrated streams, rivers, and coasts everywhere from populated to completely remote areas at such a rate that it's, you know, just, it's insurmountable to have all of the invasive mangrove removed from the Hawaiian Islands. And so the success of the fisheries going forward into the future really depends on our informed management of these areas like invasive mangrove forests. But there are some community groups that are removing mangrove at just really inspiring rates. These ecosystems that have had mangrove for over 100 years are facing an entirely new ecosystem that they haven't seen in over a century. And so being able to measure how these ecosystems change is very important, and understanding how sensitive they are to disturbances and how resilient they are still has to be determined, as this removal is very much happening now. This is also very important for the aquacultural industry, Hawaiian fisheries in general. So Hawaiian fish ponds helped to feed 1 million people, approximately 1 million people before European colonization. Compare that to the 1.4 million people that currently live in the islands today. However, now Hawaii imports more than 85% of its food, including 50% more, or 50% of its seafood. And that's just kind of ridiculous because we're in the middle of the ocean. And then compare that to globally, aquaculture produces about 50% of the world's seafood. And this is primarily due to a result of overfishing and pollution that has happened in our oceans, so that in order to get reliable seafood production, we actually have to grow it ourselves. But some of the fisheries that do occur elsewhere in the world are not necessarily very good for their environments. They overuse antibiotics, or they pump nutrients and fecal material out into the environment outside of the fishery. And so in that sense, Hawaiian fish ponds can serve as a sustainable model for aquacultural systems. The native Hawaiians truly were fisheries geniuses. They knew when to fish, when not to fish, and they knew the exact kind of conditions and environments that promote fishing. So, I mean, I guess with that, I would like to acknowledge everybody in the Alligator Lab. Thank you so much. You guys are especially essential to me, particularly in this time, more so than others. Thank you also to the Hanauma Bay Education Program for having us do this talk. Also to Hawaii Sea Grants and PICASC for funding this project and for funding me. And I guess this is a little bit different of a format that I'm used to. So I think, Maya, you said we might take a question or two as we switch over. Or if you have a question, you can put it in the chat. Usually they save the questions till the end.
If people have questions, please feel free to enter them into the chat, and or just unmute yourself and ask a question in real time over the microphone. Thank you so much, Becca. So let's switch to the next presenter. We'll have Marcus bring his slides up. Any questions for Becca?
Chat. Any questions? Maybe we'll come at the end.
Okay.
I just gotta think about them.
Yeah.
Yeah, they're thinking about good ones. Thanks, Becca. Let's, Marcus, you can go ahead and start your talk now.
Okay, got it. Okay, so thank you everyone for coming. I'm here to talk about the value of the beach. So what we looked at was an economic valuation of Waikiki Beach. And if I can, oh, there we go, okay. So I can quickly show you what I'm talking about. I'm sure many of you, if not everybody, has been to Waikiki. And you know, as a map, we can see that Waikiki has several sections, and you know, some parts are obviously larger than others, some parts are very smaller, some part is a rocky shoreline, right? Or some parts are very sandy, and people do all sorts of different activities on the beach, whether that's recreation, you know, swimming, surfing, whatever the case may be. Now, the problem that we're facing today, especially, is that Waikiki is slowly eroding away. As you may or may not have seen on the news, you know, with the king tides and the erosion and things like that, the beach is slowly getting smaller and smaller. Now, we want to think about, you know, if this is something that's happening, why is this a problem and what exactly is happening? So the beach itself naturally moves back and forth on the shoreline. This is quite typical of any beach, of any shoreline. However, in this case, Waikiki, there is a lot of built-up structures right behind it. As we can see in this photo, right? There's the beach here, you know, the waves are up all the way up. Some sandbags, right? There are hotels, there are restaurants, there are, you know, lots and lots of things right behind it. So in that sense, the beach cannot retreat, right? It does not move back and forth in this case. It can only sort of, you know, be transported away to other places. Now, we should also remember that Waikiki is an artificial beach, and as a result, it erodes more than other shorelines on Oahu. However, over the years, beach renourishment, that is, you go dredge up some sand slightly offshore and you put it back on land, has maintained the beach width over time. So over time, it has not really changed very much. So what we're looking at here is our study is focused on recreational opportunities. That is, we would like to understand the value of recreation in Waikiki, which informs and justifies any future beach renourishment work. So what is a beach day worth, and how do you determine values for things that don't have prices, right? So ever since the 1970s, economists have developed techniques to assign defensible values to the environment. Perhaps most famously, you may recall the Exxon Valdez oil spill. In that disaster, there were two groups of researchers that developed competing methods to put a price, if you will, on the environment and to value the environmental harm that occurred from that oil spill. So we incorporate the best of both of these methods that have been refined over the years to jointly estimate results in our study. Right now, we have one of these results available for the first phase, which I will be showing today. Okay, so this is the issue here. Prices don't always exist for a lot of natural resources. So for example, when you go to the beach, there's often no admission. But at the same time, I think intuitively, we know that natural resources, such as beaches, are very valuable. The problem is, without a price, we cannot easily determine how much the beach is worth and how much is it worth for whatever purpose that it may be. If you excuse the economics lesson here, if we use a very simple diagram like this to illustrate what I'm trying to say, people come from all over the place to visit Hanauma Bay. Let's use that as an example. There's an entry fee if you're not a resident. So let's call this shaded area A, right? Just the number of visitor times however much they pay in price. So this is a observable part of the value of the beach. So this we know because some people pay an entrance fee. There's also an unobservable part that is in this shaded area B. Okay, there's some additional cost to get to Hanauma Bay or the beach site in this case. People expend transportation, time, and effort to get somewhere, right? So when you go to the beach, yes, you don't pay anything, but it is in fact not free. In fact, it takes up quite a great deal of your resources to make that recreation trip. So when you sum up all these costs and you line them up, they happen to form this nice downward sloping demand curve. So we call this, these are the unobserved costs that it would take to go to the beach. So this is what we are estimating right now. So this is just to illustrate the idea. And I wanted to go over a survey design here. So the research question is, what are visitor and resident preferences and willingness to pay, that is the dollar amount that they value for Waikiki Beach and its improvements? So to do so, we have a survey design to elicit preferences and willingness to pay for changes in beach width and water clarity, because these are the most direct things that we can affect on the beach. So for example, with beach width, you can adjust that, depending on how wide it would be desirable to have them by having beach renourishment. And for water quality, maybe you have some land-based pollution controls, right? That would make the water extra clear. So these are things that we as humans can manipulate in the nearshore environment. So to do this, we had a in-person interset survey on iPads across different segments of Waikiki Beach with UH Manoa students. So if you recall very early on in this presentation, there's that map of Waikiki with different boxes, right? So we had some people survey each of these areas to get some results. So all of these surveys are, of course, timestamped, geolocated, and we control for location, weather, and subjects' socioeconomic factors. Okay, so I wanted to quickly demonstrate what we are asking people about. So here, there is a beach today. So the second photo from the left is a real photo of what the beach looks like today, okay? The beach is that wide. But if we look at the farthest photo on the left and the two photos on the right, we can see that the beach width has been manipulated. That is, we in Photoshop made the beaches wider or narrower, okay, to ask people about how they feel about the beach. So that is 50% of the beach width, 100%, which is what it is today, 150%, and 200%. So we call the beach that is there today the beach as is, okay? And then we define some alternative beach conditions that may be possible. And these are all very realistic things that can be done through management or through some sort of renourishment project. Likewise, we wanted to ask people about water quality. So for example, the beach today is the second photo from the left, and the water is typically about this clear, okay? So the numbers are not on the photo, but it's about the water clarity that we're talking about is about 10 feet, okay, that you can see underwater is typical on average. And the one on the farthest left is 5 feet, and then the two on the right are, I believe, 15 feet and 30 feet. So here, you'll notice that one of the photo, the beach today, is a real photo, okay, that we have not changed, and we've manipulated the water to become either more clear or less clear. And the reason for this will become very apparent in a minute, so let me just continue here. Okay, so in our survey, this is what we are asking people. In our survey, we ask, first of all, there is a beach today. So on the far left side, there is always a beach that is approximately what it looks like today. And it would be as wide it is today, the water is as clear it is today. And also it doesn't cost you anything to get there because we are asking people on the beach to answer this survey and they are already there, right? So there is no cost, obviously. And we ask people, they are given two alternatives. That is, there is some random combination of these changes in alternative beach 1, and a second random combination of changes in alternative beach 2. So as you can see here, in each of these, something has changed, right? Whether the beach has, maybe it's gotten wider, it's gotten narrower, maybe the water is clear or less clear, and there is typically a change in price, or maybe not. But something is different. The idea is that by asking questions like this repeatedly, we are able to get a price through statistical methods for each of these attributes, right? So we can find out how much do people value making the beach wider, or let's say you have some pollution control measures and the water is now clearer. So in our fieldwork, we've designed a survey that goes on an iPad, and we interviewed 398 subjects from November 2019 to January 2020. And of course, all results are geolocated, so we're able to tell where people are when they took the survey, right? So because the way we set up the beach width is some percent, right, of what it is today. So by looking at where people are, where they took the survey, we can have an accurate reading of what sort of beach widths they experience and what that says about what they think of other beach widths, okay? And in the survey, we cover all 7 sections of Waikiki Beach that we've identified, and we captured a lot of choice invariant variables, that is to say, things like weather, some sociodemographics, and travel patterns of the respondents. And of course, this includes both local residents and also tourists as well from a variety of different places. And here are the results. If you have any questions, I can go into a mathematical model also. But basically, the gist of what we found is that in our results, there are 2 classes of respondents. There are 2 kinds of people that generally exist that we've interviewed on Waikiki Beach. 60% is the first group and 40% the second group. The first group especially cares about the beach as it is today. That is, they like the beach just the way it is. On average, beachgoers are willing to pay about $16.48 for an extra foot of underwater visibility, and that is a relationship that increases linearly up to a maximum of 30 feet, because once you get past some point, right, it is not possible to perceive any additional clarity, right? You can only see so far, so there's a limit to that. And on average, beachgoers are willing to pay $4 more per foot, up to a maximum of 228 feet. That is to say, beachgoers would like the beach to be wider, but only up to some point, right? And here we modeled this curve as a quadratic relationship. That is to say, it increases very steeply at the beginning, okay, and it sort of levels off in the middle, and then declines. So a maximum willingness to pay exists at 228 feet. And the beach, as is, we found to be worth $547.68 per trip. And there's something we should qualify this statement with, is that this is true of the type of respondent, that is the first group, who cares about the beach as is, meaning these people really, really like the beach as it is. They don't want any changes to it. And in fact, when we look further, the type of people who belong to this group, they don't support beach renourishment. That is, they don't want the beach to get substantively larger. But at the same time, they also don't want the beach to erode. They don't want the beach to get smaller, okay? And these are typically visitors that hold this view. So there's several implications we can draw from this, okay? That is to say, beach renourishment is justified. It is very reasonable for some public effort to be made to make the beach wider. And of course, this is true in many places around the world where beach renourishment occurs. So in this case, society should be willing to pay up to $4 a foot, up to 228 feet of beach. Likewise, nearshore water turbidity should be managed, and we can justify that management. Society should be willing to spend up to $16.48 per feet to improve underwater visibility up to 30 feet. Okay, but no more than that, for at least for recreational purposes, because there is no way to perceive that typically. And there is a tremendous value for the beach as is, and any beach management work should consider very seriously how much people do not want the beach to change. And this is for better or worse. This is simply a preference that we've discovered in our results. Oh, and I would like to acknowledge our funding sources. This would include the Waikiki Beach Special Improvement District, the University of Hawaii Sea Grant, Center for Sustainable Tourism, and of course, all our student surveyors as well. So thank you. And if I can give that back to you, Maya.
Thanks, Marcus, for your talk. If people have questions, feel free to enter them into the chat box. I do have a question for Marcus. So Marcus, let's have you stop sharing your screen, and I can read the question as Honor brings her slides up. So the question for Marcus is, a unique feature of the waters off Waikiki is as a final resting place. How do you add the valuation of being a place for iwi kupuna into your calculations?
Okay, so let me, actually I don't have that chart here, but typically, okay, when we value a natural resource, right, there are many different things that you're valuing. So in this case, the sort of the first thing that people think about when they're in Waikiki is that it's a recreational site, okay? So we call this a use value. So in the total economic valuation, we say The use value is how much value people put on being able to have recreation at the beach, whether that's for surfing or whatever the case may be. So that's what they value it today for today's use, okay? There are also different values. So some people would value the beach, maybe they would like to use it in the future, okay? Maybe they simply value the beach because it exists, okay? There are many different uses. What we focused on was the use value. So this is the sort of most concrete thing that people interact with every day. So in later phases of our project, we will be looking into all the other values also as well. So these would be different types of uses, whether that's for maybe somebody would like to visit Waikiki some other time, or perhaps they simply like the fact that it exists and it is for whatever other use that there is.
Okay, thanks, Marcus.
Sorry, should I read off the question or is that a different thing?
Say that one more time, Marcus.
Should I read off the question or is that or is that coming to you?
It's in the chat.
You can read them in the chat box. So there's lots of good questions, and we're gonna hold the rest of those questions till the end. So next we have Honor Booth, and I see her slides are up. Whenever you're ready, Honor, take it away.
Aloha kako. My name is Honor Booth, and I am a graduate student in the Department of Chemistry at UH Manoa, working with Dr. Megan Donahue and Dr. Eileen Nally of the Hawaii Institute of Marine Biology and Dr. Kim Polinsky of the Nature Conservancy. Our project, Ai'ia e Kai'ao, Ai'ia e Ke Kanaka, Eaten by the Fish, Eaten by the Human, investigates land-based pollutants in coastal ecosystems and the transmission of these pollutants to local communities through seafood. This evening, I will be sharing the purpose of our project, the study site we're focusing on, including where we will collect samples and land uses in that area, the compounds of interest that we will be looking for, and the next steps for the project. So as many of you may know, Hawaii's geography has deep land and sea connections, and human activity on land affects the health of coastal ecosystems. This was true when the Ahupua'a system was thriving and has not changed despite the changes in land use that we see today. The goal of our project is to better understand how pollutants from modern human development are transported to coastal ecosystems, and in some cases, back to the community through fish consumption. Right now, we are focusing on West Loch, our study site in Keawalu o Pu'uloa, or Pearl Harbor, on Oahu. West Loch is subject to stream inputs from 3 ahupua'a in the Ewamoku, Honuliuli, Ho'ai'ai, and Waikele. We are currently in the planning stage, coordinating with the groups responsible for our 2 target collection sites. Pohala Marsh, which is under the responsibility of the Department of Land and Natural Resources, Division of Forestry and Wildlife, and Kapapapuhi Point, which is under the responsibility of the nonprofit Malama Pu'uloa. Pohala Marsh is at the Waikele and Kapakahi stream mouths. The top picture is of the marsh, which is home to native birds such as Ai'o and Alai'ula. The bottom picture is of the remains of the bridge over Waikele Stream, which when we did reconnaissance, we actually found out that that bridge is going to be taken down and they're going to continue building the bike path that goes all the way around Pearl Harbor. Kapapapuhi Point is at the Honuliuli Stream mouth, and the top picture is of where Honuliuli Stream enters Pu'uloa through the West Loch Golf Course. The bottom picture is of the waters of Pu'uloa through the mangrove along the shoreline. And both of these sites were chosen for their proximity to stream mouths and also because of future mangrove removal projects. So as mangroves can trap sediment, removal of the mangrove forest will release sediment that could be holding the residues of organic pollutants and heavy metals. The breadth of the Ewamoku allows for different modern day land uses, including modern and historical agricultural use at the base of the Waianae Range and further down in the Ewa Plain, residential and industrial uses along the Waikele stream, and recreational uses such as golf courses along the coast. Due to the different land uses in the Ewamoku, we expect to find a diverse set of known pollutants. Some of our compounds of interest include organic or carbon and hydrogen-based compounds such as polyaromatic hydrocarbons, polychlorinated biphenyls, pesticides, and ultraviolet filters, as well as heavy metals such as arsenic, lead, nickel, and zinc. Polyaromatic hydrocarbons are compounds that are characterized by linked ring structures with double bonds. These compounds are created through the combustion of organic materials such as gas, wood, and garbage, and are often used in the production of dyes for textiles and plastics. Some examples of polyaromatic hydrocarbons are anthracene, which is used as a precursor for the red dye alizarin, naphthalene, which is a known human toxic that is used as the ingredient in mothballs, and pyrene, which is a byproduct from forest fires, cigarette smoke, and charred food. Anthracene, naphthalene, and pyrene are all known carcinogens in fish and humans, and are among the polyaromatic hydrocarbons that are regulated by the EPA under the Clean Water Act. Polychlorinated biphenyls are compounds that are characterized by bridged ring structures with double bonds that have attached chlorine atoms and are used in pesticides, plastics, electronics, and machinery. Animal studies have shown that PCBs are carcinogenic and can negatively affect the immune, reproductive, nervous, and endocrine systems. Human studies further support that PCBs can cause cancer. Due to these adverse health effects, all PCBs were banned from use in the United States in 1979. However, their stability in the environment can cause them to persist decades after their use. Pesticides are used in agriculture, landscaping, and golf courses. While DDT is a famous pesticide that has been exhaustively studied and quantified, the story of this compound is an important reminder why the impacts of pesticides should be thoroughly evaluated before being made available and used in agriculture and the environment. Used in the 1940s to control insect-transmitted diseases, trucks like the one seen in the top right picture were driven through neighborhoods to spray DDT and kill mosquitoes. So my mom has told me stories about the DDT trucks that used to drive through her neighborhood in Kaneohe when she was growing up in the 1950s, early 1960s. And she told me that, so my grandparents are both from Wailuku and Maui, and they grew up around agriculture, plantation, and they knew what pesticides did. So they pretty much would tell my mom and all my aunties to get in the house, lock up all the windows and doors, and they wouldn't go outside for up to an hour. So that's just to give kind of a perspective of how close the usage of this pesticide is to home. Rachel Carson's 1962 publication, Silent Spring, began to raise awareness of the dangers of pesticide use, and DDT and its breakdown products CDE and DDB were found to have adverse effects on wildlife, such as the thinning of eggshells as seen in the bottom right-hand picture. Although EPA banned the use of DDT in 1972, its breakdown products are highly persistent and can still be found today in areas that were exposed to the pesticide. Organic ultraviolet ingredients in plants, but are also added to plastics to prevent damage from sunlight. As plastic debris moves through the environment, it can leach UV filters and their breakdown products. While the exact effects of UV filters from plastics on biological systems is still being evaluated, certain UV filters from plastics have been shown to accumulate in the fat tissue of aquatic and marine organisms, and even in humans. A limitation in detecting UV filters from plastics in the environment is that most plastic formulas are proprietary. So you don't know what UV filter is being used in a certain plastic formula unless you actually analyze its chemical structure. As plastic debris has become ubiquitous in coastal environments, quantifying the presence of UV filters from plastics can elucidate the extent of plastic pollution in the environment and inform toxicology studies by providing known environmental concentrations. Heavy metals are known toxins that can enter coastal ecosystems naturally and through human activity. The presence of certain heavy metals usually depends on local geology. However, activities such as mining and excavation for subsurface infrastructure can release these elements into waterways and topsoil, where they're easily transported by runoff to coastal ecosystems. Heavy metals can also be products from industrial operations and are used in tires and anti-fouling paint. Arsenic, lead, zinc, and nickel are of specific interest, as past studies by Dr. Nally and Dr. Donahue have found threshold effect levels where negative effects on aquatic life is probable of these heavy metals in certain fish. Furthermore, while nickel is a common element found in Hawaii's soils, unusually high concentrations were found on the reef flats where these fish were collected. Heavy metals can be toxic as they displace other metals that animals use for regulating biological functions or making proteins. For example, lead can displace calcium in osteocalcin, a protein responsible for bone mineralization. As lead makes a stronger bond with the protein, it blocks calcium from binding to the protein, preventing calcium from being added to bone tissue. So the next step in our project is to continue to build our relationship with different stakeholders in the Ewa Moku, including the United States Navy, the Department of Land and Natural Resources, Division of Forestry and Wildlife, and Malama Pu'uloa. From there, we will begin planning the collection of water, sediment, and fish samples. With our results, we hope to raise awareness of the risk of fishing in certain areas and inform restoration processes to ameliorate conditions for future aquaculture and conservation projects. And with that, I'd like to say mahalo piha to Hawaii Sea Grant, HIMB, Dr. Donahue, Dr. Nally, and Dr. Lensing for the amazing opportunity to participate in this research project, to our community partners from the U.S. Navy, the DLNR Division of Forestry and Wildlife, and Malama Pu'uloa, especially Sandy Ward from Malama Pu'uloa, for allowing us to do work in their areas. And then also, finally, to the Leeward Community College Kilo Aina Hui for compiling the secrets of Pu'uloa, which has been an incredible resource for me, learning about the history of Pu'uloa. And finally, to my family and friends for all of your support. And then also mahalo piha to all of you for attending my talk. He mau ni nau ka oko. Do you have any questions?
Thank you, Honor. Great talk. Okay, what we'll do now is if people have questions, please do enter them into the chat, and we're gonna bring back Becca and Marcus as well. And maybe what we can do, normally if we were in the room together, there would be applause, and I see people doing the clapping in your boxes. So applause and thank yous for our awesome graduate researchers who gave great talks tonight. Thank you so much for presenting your work. Okay, so I do see a question for Honor from Ku'i. Ku'i, would you like to ask the question yourself, or do you want me to read it?
No, I can ask it. Hi, Honor. Hi. Okay, so are the same chemicals that are in pesticides used in herbicides? And I only ask because I think they recently banned it in Kona, but I know Kona was spraying herbicides along their roadways for like years. And Kona is a heavy fishing town. So yeah, I'm just wondering, are herbicides the same as pesticides?
So herbicides are in the group of pesticides. So pesticides address herbicides, insecticides, fungicides, pretty much things that kill what is considered a pest. And so with your question in respect to what was done in Kona, that really just depends on what specific herbicide they banned. And it's kind of interesting with like the chemical industry, when something is banned, they ban a specific chemical. So like one of those specific structures that I showed, a chemical company can go in, and when they synthesize one of these molecules, they can add in like an extra methyl group. So that's a carbon with 3 hydrogens attached to it. They can add in one of those somewhere where the molecule still has its same function, but because of this extra methyl group, it is considered a different molecule and is no longer under the auspices of the ban, and then can be used. So so even if like an herbicide is banned, there's loopholes to get around it, unfortunately. So yeah, I hope that answers your question.
I see a hand up from Bob. Bob, would you like to ask a question?
I do have a question with regard to metals that can be toxic or be pollutants. You had down lead and nickel. You know, I was reading an article today about whistles that are coated with nickel. So I have to assume that nickel in some state is probably not toxic. And when you look at all the lead weights that are used by fishermen, is the lead inert, or does it have to oxidize and get combined with other things before it becomes toxic?
That's a really good question. So I am an organic chemist by trade, so I know more about you know, organic molecules than metals. But I do know that certain metals need to be in a specific oxidation state to be able to be active biologically. So with respect to lead, the main things that I know about it is how it gets incorporated into organic molecules and then is transported into humans or any kind of biological system. So unfortunately, I can't completely answer your question, but yeah.
Thank you.
So we have a couple questions for Marcus. If Aka wants to ask it, or I can ask it for you.
Oh yeah. Hello, Marcus.
Well,
Yeah, thanks for your talk. I was just wondering, you know, just because of the sampling that you've done, it was focused around the Waikiki area. I mean, that makes sense because that's your target of what you want to learn about more. But I mean, I was wondering if you know, people that aren't in that vicinity, in that locality, you know, they might have thoughts also about the valuation of places, and maybe it might be good to have a broader survey.
thank you for the question. So what happens when you survey people at a particular site, okay, and this is, in recreation, this is very commonly justified, is that even though by surveying people at a particular site, you incur a on-site bias, okay, because if you think about this for a moment, everyone who answers the survey must be going often enough that they happen to run into a surveyor, right? So if you think about that, you're more likely to encounter people who, I mean, yes, there are some people who happen to be there, but also the people that you run into, if they are, say, local residents, then they would be going more often. So we also we also have to perform a adjustment for the on-site bias. So that's the first part of the question. The second part would be that in our additional analysis that we are doing, we also switch to a model where we are agnostic about where people are interviewed. That is to say, we are able to, so for example, we give a survey, the person can be answering that survey anywhere, right? And then we would be able to determine, we would be able to ascertain some values for the different features of the beach. The main, I want to bring up one thing, is that the main reason why we had a specific location for each of the respondents is not so much that it's not so much for, you know, that they are in Waikiki, but rather it's to determine an accurate beach width, because if they happen to be at some segment of Waikiki, right, the beach width varies from different segments. So that is actually, for us anyway, just a way to get that width accurately in there. So I hope that answers the question.
Yeah, mahalo.
Thank you.
So Becca, Maya has a question. You want to ask him, Maya?
Sure. So this is a question for Becca. Thank you so much for your talk tonight. I'm wondering how stable the microbe populations are throughout the year. Do they change at different times of the year when we get more or less rain? Feel free to correct me if the second part is wrong. It's not really a question, but you mentioned that we might expect microbes to change between places that have been restored versus not restored. And I can turn that into a question by saying, did I get that right? So that's my question for you, Becca.
Okay, so to answer the first part of your question, how stable are microbial communities? It's a great question. And my gut answer, well, my answer straight off the bat is that there is a lot of variability in microbial communities, especially when we talk about just the estuary system where I'm used to studying, right? So depending on how deep you go into the sediments, and even then, night and day, sometimes microbial communities will actually shift in different members, can't even see my hands, different members, as they kind of like, they come up at night to breathe, and then they kind of settle back down as the anoxic layers shift during the middle of the night. So even they change during the day, and they can also change with tides. So incoming tides, you'll have a much more marine influence in that community. Outgoing tides, much more freshwater. And yeah, we've seen during large rain events. So for example, if we have tropical storms or just large rain and heat events, we've actually been able to see and measure how the communities in the fish pond, for example, do change over time. So that input can like wash out a lot of that signal, which is why sampling microbial communities is such a tricky matter, because it is very dependent on weather. So we have to go at the right time, at the right tidal cycle, usually during the right moon as well, because moons impact the degree of tide. So we usually like to go during ole moons, which is when there's the least difference between high tide and low tide. To address the second part of your question, we do, or I guess one of the hypotheses that we are testing is whether or not there is change in restored forests. So we would expect that as they cut down the mangrove, usually they chop it right at the root level, and they burn what's on top, and then the root structures stay. And so it might be possible that the microbial communities take some time in order to change, because the root structure is very much kind of held intact. But as the mangroves are cleared away, you get improved water circulation and air circulation. You get more sunlight perforation through that what used to be the top layer of the mangrove forest, which will promote entirely different types of microbial communities if you add sunlight and if you add oxygen. So we do expect to see pretty significant changes.
Awesome answer. Thank you, Becca. So it is 7:30. We welcome people to stay online with us. If you have burning questions that you want to hear our speakers answer, please stay with us. If you have to go, thank you for joining us tonight. Let's see. Are our graduate researchers okay with answering a couple more? Because, like, for example, I see Brandon has his hand raised.
Bob has a question too.
Bob has a question too. There are several other questions in the chat.
Mm-hmm.
Are the researchers okay staying to answer a couple more?
Yeah, looks like they are.
Okay. We might have to just do those two and then we'll stop there.
Yeah.
So, Brandon, would you like to ask your question?
Sure. Aloha. This question is for Becca. Becca, I was just curious as to what sorts of microbes might be able to break down mangrove detritus?
Typically, when we talk about the microbes that are capable of breaking down the very complex lignocellulose structures, we are typically generalizing it to, there are two families of bacteria that will do it, and there's also white rot fungi will typically also break that down. However, just because we have those members present, like if we find them in our DNA sequencing, that doesn't necessarily mean that they're doing it. So microbes have this sort of weird thing where like they can just exist and not be really interacting with their environment. So just because we find the rot there doesn't necessarily mean that they're eating the mangrove on a regular basis. And so that's why it's important that we're asking that sort of that two-pronged question.
Wow.
Okay, Bob, last question of the night. Let's hear it.
I'm glad that you're studying an area and studying it thoroughly so we can get insights relative to how it all works. My concern is that there are a lot of areas in the islands that don't get that kind of attention. We have an awful lot of government and organizations that there's the State Department of Health, there's the Clean Water branch, there's this branch and that branch, and it kind of goes on and on. But I feel like there is a lot of pollution, but without data, you know, you're sort of a voice in the wind. How, who, what, how can we reel this puppy in and make a difference long term? That's for you, Honor.
Honor.
Hi. You know, with the Department of Health and making, actually I was an intern for the Clean Water branch for the past year, and I still volunteer for them, so I know how all that stuff works. With Department of Health, they require an enormous amount of data before they will make health recommendations. So really, what it comes down to is more of a bottom-up approach, I guess. So using different methods of communication to pretty much let the community know, you know, what we're finding. But also, you have to remember that you know, some of these communities, they fish because they have to. They fish because they don't have the financial option of going to the store to buy food. And for a lot of them, it's between the choice of fishing and providing food and putting food on the table, and not fishing and not eating. So you get into a really difficult position where you want to share your results, because people need to know these things and need to know the risks of fishing in these areas that are highly polluted. But at the same time, you know, telling a community you can't fish here anymore displaces that community's resources. They have to go fishing in another area where there's already people who are fishing, and takes away from, you know, this whole idea of like an ahua has everything that you need to live. I hope I'm answering your question, but from what I can see from like my experience working with ultraviolet filters from sunscreens, is a champion. You know, we need someone who will all of the data, preferably someone who's actually not a scientist, because I feel like that takes away from the unbiased character of the results in and of itself. So you need someone to take this data and push it forward and, you know, show it to lawmakers, and so that things can be changed. Ooh. I think I might have froze a little bit over there, So you need somebody to compile this all together so that things can be changed from a top-down approach as well. And that just means more research. And yeah. Did I answer your question?
It was a good answer. I wish the government top-down could take care of it. It's sad to me that so much fundraising has to go into nonprofits to go out and collect data and to try and help the government do their job. It would be a lot. I'd like to see all that fundraising go to other sources. And the only reason they have to do it is because the government doesn't do it, and it's frustrating.
For me, I think bioremediation is a really good solution for a lot of these things, and using invasive species for bioremediation, but that's like a whole nother branch of chemistry and biochemistry. That's like after I get my PhD. But yeah, thank you.
Okay, thank you everyone so much for joining us tonight. Thank you for all of your thoughtful questions. And we're gonna close the seminar. Thank you again to our graduate researchers who gave great talks. Please join us next week. We'll have 3 brand new talks to show you, same time, 6:30 p.m. And yeah, please register to join us next week. That's it for today.